Well, we're moving through our process of finally extracting the DNA. This is the last step in that sequence, isn't it, Mary? We're going to heat our cells that have been purified with the matrix earlier, and we require 2 temperature regimes to bring about that extraction. Yeah. Okay, so could you just show us then, Mary? We won't go through the whole process because it takes probably about 15 to 20 minutes all in total, but we will just quickly show you, and then I think it's quite obvious.
Okay. So first thing you need to do is you need to make sure that you mix the Instagene beads with your sample, because they're going to remove all the ions that come out of the cells as you break open the cells. Now, you need to float your samples in water that you've got at the right temperatures. 56 degrees centigrade is to denature the enzymes that are in the cells, so that when you burst open the cells, the enzymes don't break down the DNA. So you need to put your samples into something that will float in the hot water. This is just a sponge.
Yeah, just quick ready idea, yes.
Make sure that the cells, the part with the cells, is underneath the sponge, so that when you put the...
Okay, so 56 first, is that right?
56 first, so that when you put the samples into the hot water at 56, it is actually the cells that experience the hot water. And you time 5 minutes.
Okay, so we're going to fast forward. In 5 minutes' time, what do we do, Mary?
After 5 minutes, you take out the samples and you flick to mix again.
Okay.
And then they go back in for 5 more minutes.
Okay, so we've had 10 minutes with a flick halfway through.
Yeah.
Okay, so now assuming we've completed our 10 minutes total at 56, denaturing the enzymes, as you said, we're going over to...
You flick again, you take them out.
Okay, right.
Yeah, you flick the contents again to thoroughly mix the beads, because remember, every inhibitory ion that is produced has to have a bead to remove the ions which are a bead to remove all the ions which could otherwise stop the rest of the PCR process.
Okay. And we're not going to put them in because we're going to do this for real, aren't we? So this is just a demonstration that those beads would float in there at 100 for how long do we reckon?
5 minutes.
Another 5 minutes.
So it's 10 minutes in total at 56, and then 5 minutes at 100 degrees centigrade.
So we're going to put them back in and do it properly ourselves off camera, and then come back to it a little bit later. Okay, thank you.
Thank you.